So hi guys, let's begin with our second chapter. And okay, just wait for a second. Okay, so let's begin. This is our second chapter, which is General Biology, in which we will study germ, cell formation and fertilization, prenatal development, induction, competence and differentiation, formation of the three-layered embryo, formation of the neural tube and fate of the germ layers, folding of the embryo and neural crest, which is the most important part of this chapter, the neural crest part. Okay, so let's begin. In the first paragraph, summary of this whole thing, the germ, cell formation and fertilization, in which we have the somatic cells and we have the body cells. Somatic cells are actually contains 46 number of chromosomes which are all diploid. That means there is mitosis here, okay? And then we have these 22 homologous chromosomes which are autosomes, okay? Which are paired autosomes, and one pair is for the sex chromosome, and sex chromosomes are the X and Y, which are the XX and XY, okay? I hope you are getting it, okay? So as we all know that this is the most complex course of dentistry, but it's okay. Take a deep breath and study with me. Okay, so we have this confusion thing here. As you can see that there is 46 chromosomes, okay, that we have already discussed here. And then we have one pair which is autosome, okay. One pair studies sex chromosome, XY and XX, and then we have this fertilization process in which we have male and female gamete fertilized, fused together, okay. And the spermatozoa and ova. Ova is for the female germ cell, and spermatozoa is for the male germ cell. So they together fuse together to form a zygote. After the formation of zygote, they make their number haploid. That means haploid, okay. That means there is meiosis here, okay. And okay, so just forget about all. This is the basic concept of this whole paragraph, okay. If these mitosis, meiosis and whole thing, okay. Okay, so there is most important thing here, like diseases. Clinical stuff is very important in our Tencates, okay. So do not forget to learn all these diseases like monosomy, Down syndrome, and Down syndrome again, and then there is in Down syndrome there is shortened palate, protruding and fissured tongue, and delayed eruption, and 10% of all human malformation are caused by an alteration in a single gene. So okay. And then we have the diseases are actually divided into autosomal dominant and autosomal recessive. On the basis of the dominant inheritance and the autosomal recessive inheritance, we will discuss a few diseases, okay. For example, we have these, okay. We have these achondroplasia, cleidocranial dystosis, osteogenesis imperfecta, which is related to bone obviously, and cleidocranial dystosis is actually related to the muscles, okay, if I'm not wrong. And there is the dentinogenesis imperfecta, which is the second layer of the teeth. Okay, we will discuss it in detail. Dentinogenesis imperfecta 1, imperfecta 2, because it's really important in from post-OSPE point of view. Okay, and then we have the letter 2 condition. Okay, dental hard tissues. Okay, what is dentinogenesis imperfecta? Dentinogenesis imperfecta is actually caused by the mutation of the dentine sialophosphoprotein gene. Okay, this gene is really important. Okay, so anyways, you have to memorize this by heart. And then we have this diagram of dentinogenesis imperfecta, which is autosomal dominant disease, which is caused by the sialophosphoprotein. Okay, and then we have the examples of the autosomal recessive, which includes chondroectodermal. Chondro means ear bone. Okay, the soft ones. Ectodermal, which is the outside, dysplasia, abnormality. Okay, and then we have microcephaly, means short face. Okay, cephaly means head. Okay, cystic fibrosis means there is soft muscles. Okay, fibrosis comes from fiber. Okay, and then we have fibrosis. After that, we have these genotypic and phenotypic ratio. Phenotypic is basically the physical defect, and genotypic is the gene defect. Okay, which is genetically present. Okay, and then we have this teratology. Okay, the teratology thing. Okay, this teratology. What is teratology? Teratology is the study of developmental defects that I have already mentioned before, that there are some autosomal recessive defects and there are autosomal dominant defects. So these are studied. These are all defects which are studied under the subsect of teratology. Okay, so let's move on to prenatal development. Okay, this is the chart for the prenatal development. I hope you can see it. Oops. Yeah, I hope you can see it. So there is proliferation and then migration and oh my god, fetal, embryonic, and then these are fertilization, cleavage, blastocyte, implantation, bilaminar embryonic disc, and all. Okay, first of all, there is induction, competence, and differentiation. Okay, let me tell you this whole important thing about this diagram, that there is autocrine and paracrine regulation. Okay, these are the two cells which are autocrine, means they are forming automatically. Okay, automatically. And there is, whereas paracrine, target cells, which is not formed automatically. Okay, I hope you understand it. Okay, so there is the three-layered embryo. In three-layered embryo, we have this ectoderm, mesoderm, and endoderm. Okay, ectoderm, endoderm, mesoderm. Ectoderm is the outermost layer, endoderm is the innermost layer, and mesoderm is between the outer and the innermost layer. Okay, I hope you understand it. Okay, so formation of three-layered tissue. Okay,